Hello.
Hello, Nitin. This is Lakshmi here.
Hi, Lakshmi. How are you doing?
Yeah, doing fine, Nitin. Good morning.
Very good morning to you also. So I just wanted to see if the slides are going to work fine.
Sure, you can share your screen, I think.
You can see my screen, right?
Yes. Yep.
Can you see the video or no? Can you see
It is.
something moving?
Yes.
Slide to check. You could see the image, right?
Yes.
See the movie.
Yeah.
To check. All right. I hope you are good to go.
Yeah.
I can
We'll just
stop.
wait.
I think I'll stop sharing and then.
Yes.
Hi Nitin.
Hi Ravi, how are you doing?
Fine, fine. I see that lockdown impact has happened on you.
Yeah, yeah. Yeah, so
No, Hi Lakshmi.
Hi, Ravi.
Hi everyone. No, Nitin, I really thank you for your taking your time, Nitin, and putting this together. It's really nice. We have had some good feedback so far.
Thank you very much, Ravi. I think this is a very wonderful initiative, and you or someone else should keep it going. And I'm very happy to be part of this. I hope I could do justice to the.
No, I think we are all looking forward because something like this has probably not happened, and we're recording and keeping all this. So I think anybody can listen to this. People, although we had limitations over participants because of when we got the grant from DST, they were clear about number of participants. But through this mode, we can have many people actually. So I'm hoping that will be useful, and let's see if it is really beneficial to students. We could have more sessions slowly. But what I'm saying is that it's not just like giving a regular talk, and all the speakers and their students had to work together to put a data set and go through teaching. A lot of people have put a lot of effort, so it's very nice to see that.
Yeah.
So how's IIT Kanpur doing?
IIT Kanpur is slowly coming back from the lockdown impact, I believe. We had to send all the students back home in April.
Yeah.
And now few batches, PhD students, few of them have come back.
Very nice.
Yeah, so probably by mid-September, most of our students will be back, and hopefully,
very nice.
hopefully we don't have another situation to send them back again.
No, that's the fear everybody is talking about, as if it was lurking around the corner, but we cannot see it yet, kind of.
Yeah, you never know these things like. And Kanpur is a very different situation altogether because the medical facilities available in the city is probably not as efficient and great as in Bangalore. So taking the risk of
everywhere it's
accommodating the students in the
I know everywhere it's overwhelming, but I think you guys took the right decision of sending all the students back home at that point of time. You have avoided a big issue, no? Anyway, really nice to have you here, and I'm looking forward to hearing you. And so I think the time is up, and we should not delay. In my also, all the credit goes to my students. They're the ones who's driving this. So somebody set up to introduce you, so they will go ahead and do it. So welcome once again.
Thank you very much, Ravi. Thank you very much. Yeah.
Thanks.
And it's my
Akshaya?
pleasure being here.
So Lakshmi ma'am will.
Yeah.
Lakshmi ma'am, please.
Yes.
You can go
take
ahead
it away.
with introduction.
Yeah. Good morning, everyone. I once again welcome you, Nitin, and thank you very much for taking your time for this session. And we will be having good things. So before going into the session, I would like to introduce Dr. Nitin. Nitin did his master's in physics from Mahatma Gandhi University, Kerala, and pursued his PhD and moved to Taiwan to do in nanoscience and technology, where he got trained to design and develop optical microscopy such as confocal and turf. And he also optically characterizing fluorescent nanodiamonds at single particle level for the bioimaging applications. And the interesting part I find in his profile is he didn't stop there. He took it and in his postdoctoral training in France, where he tried to apply his learning from the PhD, where he tried to track the single endosomes with the fluorescent labeled fluorescent nanodiamonds in the distal dendrites of primary neurons to identify genetic factors involved in trafficking dysregulations associated to neurodegenerative disorders. And later he worked in ICFO Barcelona um, to master a single molecular localization based super resolution techniques such as POM, STARM, and their applications in understanding the intracellular transport. And I see then he moved and joined as associate professor in IIT Kanpur, where his research group currently focusing on developing advanced fluorescence nanoscopy techniques to study the molecular interactions and dynamics in cellular processing related to nutrient sensing, intracellular trafficking, and neurodegenerative disorders and lysosomal storage disorders. I hope this is a wonderful work I'm seeing in your profile, Nitin. Looking forward to your session. I think the coordinators will have a couple of instructions to the participants. Over to you, Kirti.
Hello everyone. We have two small instructions for the participants. We request all the participants to keep their audio and video off during the session. And if you have any questions or doubts during the session, you can type in your questions and we will convey the questions to the speakers. Over to you, Dr. Nitin.
Thank you, Lakshmi, for that wonderful introduction, and thank you, Kirti, for this housekeeping that you're doing efficiently. And thank you, Ravi, and his entire team for inviting me over for this wonderful initiative that you have taken up. So imaging is not just about pretty images. It can definitely give you quantitative information. And I'll be talking about how you deduce a little bit in that line, like how you deduce or decipher this quantitative information out of these pretty images that you might get if you are an expert in imaging, right? Imaging expert doesn't mean that you take pretty images. You also try to put some numbers onto that, on those images that you have taken. So yeah, so let's start. And so today I'll be talking about cellular logistics, like how the protein is transported from one location of the cell to the other. And behind this title and my name, you can see a movie being played. So here you can see mitochondria, lysosomes, and other endolysosomal compartments in motion, right? So they look nice and they look so intriguing. Why they move, where they move, and what factors contributes to their movement. All these questions just pop up in your head when you see these movies. That's a amazing thing about microscopy, that you can visualize these tiny nanoscale objects with high-end microscopy techniques. So I'll be briefly covering why what is this logistics, that cellular logistics is, what do you mean by that, and what are the different advancements in microscopy, particularly in my lab, that we do to understand these tiny objects moving. Okay, and what are the challenges associated to it, and how we plan to overcome those challenges. That'll be the major aim of my talk. So moving on, So when we talk about protein movement or cellular trafficking, before that we can draw some analogies with the way that we commute in our daily life, right? So here you can see a two-way trafficking. Some of the cars are going towards the city and the others are coming back from the city to the suburbs, probably. So this is how you commute on a daily life. Before the pandemic hit you, probably you were traveling a lot. We were all traveling a lot, and we have definite traffic rules that enables efficient trafficking, right? That we have the green signal to go, the red signal to stop whenever required. We have the heavy vehicles going on in one kind of tracks where the light vehicles move on the other. So everything is well organized and sorted and regulated such that we avoid collisions, accidents, and we can move from our location of origin or our residence to our location of function, that is our offices or your research laboratories where you can do your job, right? Similarly, if you think about cells also transport all these proteins and other organelles within to different location of their function from their location of origin. So most of the proteins are packed up, once they are produced from the mRNAs, they are packaged into so many vesicles in the ER or in the Golgi, and then they move. Suppose the protein that you want is the protein that is produced is a receptor molecule, and it has to go to the cell membrane, right, the plasma membrane. So it has to traverse through the entire cytoplasm to reach to the plasma membrane. So if you think about it, the cytoplasm is really complex, right? It is very crowded environment. So a protein of the size probably 1 to 2 nanometer has to traverse a distance of about tens of micrometers to reach to the cell surface, right? Or cell periphery. So a protein has to traverse almost 10,000. The size of the protein and the distance it has to travel is almost of the order differences of 10,000 nanometers, right? So you might not think that diffusion is a very effective way to do that, correct? So because it will be very inefficient, cell has to produce a lot of energy to do that. Probably that is why cells came up with this fantastic transport machinery that involves microtubules, these green structures that you see over here, and the motor proteins. So the motor proteins just bind to all these cargoes, which can be endosome, lysosome, pigment granules, secretory vesicles, mitochondria, all these organelles, and the other proteins that are packaged into vesicles. All these vesicles and organelles recruit motors, cytoplasmic dynein and kinesin motors, onto them, and then use these microtubule as highways or roads to walk over and then reach to the destination that they would like to. So here I'm showing a small animation. Here you can see a vesicle that is moving with the help of a motor protein. It's a kinesin here. It's literally walking on the microtubule. So you all probably know about this, that the motors use the energy from ATP hydrolysis and convert that chemical energy into mechanical forces to move the cargo from one location, that means their location of origin, to their location of function. So having all these efficient mechanisms, cell can, every cell in our body is right now doing this and making us all function very effectively. But coming back to this, the analogy that I draw of the way we commute in our daily life, we know that this was a dreamy situation, and the reality is more or less like this picture. If you have ever driven in Kanpur, you would experience this every day. So that, either the rules do not exist or people do not understand the rules. So what happens is that there is always a traffic congestion. It can be even leading to traffic accidents, and it can be detrimental for your life. Similarly, we would like to understand what are the trafficking rules within the cell. cell, again, cytoplasm is very complex and complicated and very dense, and the microtubule architecture is going to be very dense. There are thousands of proteins being produced every minute, and it has to be trafficked. So the trafficking is quite heavy, and we can expect it to be highly congested within the cell too. So what are the rules that make sure that there is no much traffic jams involved? And if these rules are broken down, or if these rules are just not being followed, do they lead to any sort of dysregulation, such as neurodegenerative disorders or metabolic disorders? This is what our lab is primarily trying to address. Now, again, coming back to the analogy that when traffic rules are not followed, right, like we always end up having situations like this. These are real pictures, right? Like this is a baby suddenly found in the middle of the road somewhere in the US. And there was a story going behind that. You can always look it up in the internet. But the question would be like, from a picture, you would only have a lot of questions. Like, how did it happen? Who is at fault here, right? And what could happen to the baby the next moment? All those questions would come with an image. Similarly, if there is an accident like this, we'll always wonder, with the picture, you will not get it. You know something has happened, but you would never know what has happened and how did it happen and who is at fault and who is to be blamed or who is to be filed the petty, right? So the solution to this would be looking into the CCTV footage, which gives you a movie rather than a picture, and that would let us know what happened, what would have happened to the baby, whether it survived, how did it eventually end up there, or who is at fault in this accident. All those things would be much clearer to us if we look into a video rather than to a pictures. So that is why, again, in understanding cellular trafficking, we rely on live cell imaging with the help of fluorescence microscopy. Okay, so here you are seeing an epithelial cell, African green monkey kidney epithelial cells, where the microtubules are in green and the lysosomes are in magenta. You can see the lysosomes moving back and forth. So the anterograde motion, or the movement towards the periphery of the cell, is facilitated by the kinesin motors, whereas the retrograde movement is facilitated by the dynein motors. Okay. These movements become much more complex than what we have already discussed in specialized cells or differentiated cells like neurons. Why? Because the proteins that are produced somewhere here in the cell body next to the nucleus has to traverse through these specialized features like dendrites and axons to reach to the synapse, right, where the neurons form connections. Now, if you think about the dimension of these features, like, for example, the axons can range from centimeters to meters in certain neurons in our body, right? And the dendrites are also in that order, like several centimeters, millimeters, and centimeters. And the proteins that are getting trafficked have hardly like few hundreds of nanometers. So you can see that they have to, the distance that they have to travel is almost 1 billion times more than their own size. So they rely particularly on the motors and the microtubules to move forward and to facilitate that two-way trafficking through these very long structures. And I have written a lot of things like map to acetylated microtubules and D-tyrosinated microtubules. We will come to that later on. But for the moment, I just wanted to give you the context that the trafficking is done through the axons and dendrites are really challenged by different factors within the neurons, and it is probably very challenging for the cell to maintain certain trafficking regulations, and then without any dysregulations happening in there. The other day, you might have listened to Professor Deepak Nair from IASC, where he discussed about the synapse itself, right? The pre-synapse and the post-synapse, and how the receptors on the post-synapse has to be organized into a specific way, or the cluster formation, such that the dynamics, the spatial and the temporal organization of these postsynaptic receptors, or the neurotransmitter receptors, are going to affect, if they are not properly regulated, then it is going to have implications in neurodegenerative disorders like Alzheimer's. Similarly, if this trafficking is dysregulated by any means, that might also implicate in neurodegenerative disorders like Alzheimer's and Parkinson's, and even in psychiatric disorders. So how sensitive is this trafficking to these such kind of neurodegenerative disorders, right? So that's one thing that we wanted to look into. So this picture Deepak might have already shown you. So here you see that there is a healthy brain on your left, and an Alzheimer's patient's brain is depicted on the right of it. And then the Alzheimer's patient brain has amyloid plaques, and then within the interstitial sites, and then within the neurons there are these neurofibrillary tangles. So what causes these neurofibrillary tangles? The fibrillary tangles are produced by this protein called tau, which associates to the microtubules. Okay, the tau binds to the these are microtubules within the axons, and the tau binds to the microtubules in the axons. And it stabilizes, its role is supposed to be stabilizing the microtubule. It might have many other roles which are still yet to be deciphered. But what we know is that if tau binds to microtubule, it stabilizes the microtubule. And tau has a very dynamic association with the microtubule. It's not always bound to the microtubule. Depending on its phosphorylation states, it can bind and unbind from the microtubule. So if it is hyperphosphorylated, these tau molecules will just be not binding to the microtubule. They will be very much cytoplasmic. And if hyperphosphorylation of tau can lead to aggregation of these cytoplasmic tau, forming tangles of tau proteins, which is called neurofibrillary tangles. And this neurofibrillary tangle presence is observed in the Alzheimer's patients. So the question that comes here is that if a tau, even in a healthy patient, a healthy person, if the tau is binding to the microtubule, what happens to the trafficking basically? That a vesicle was being trafficked by a motor protein. As the motors interact, or it sees this tau, whether the tau is going to pose a hindrance for the motor, whether it is going to ask the motor to stop for a while, or is there a very good dynamic association between the motors, the microtubule, and the taus? They are all in harmony such that when the motor arrives, the tau just detaches and gives way for the motor to move forward. Is that the scenario? We don't know, right? And whether, if such a balance is broken, whether that would lead to all these neurodegenerative processes. So what we want to understand is like whether trafficking dysregulations can be implicated in protein aggregation that might lead to neurodegeneration. Right? The other thing is like these plaques and fibrillary tangles, you will be able to observe them only after they have grown to a certain size. And by then, the patient's brain is almost towards the end of the disease, right? Like most of their neurons are already gone, and the brain has degenerated to a good extent. So it's almost at the very later stage of Alzheimer's that you can know this. But if the molecular trafficking is much sensitive to this tau protein, or for example, or for that matter, any other microtubule associated protein, and if these protein aggregates would eventually lead to neurodegeneration, can we use the intracellular trafficking of vesicles itself as a readout for an early detection of these protein aggregates or not? That's what we wanted to know. So how sensitive is the protein trafficking to protein aggregation? That's what we wanted to understand. For that, we devised an assay where we would isolate the neurons from mouse embryos, and then we would treat them with these fluorescent nanodiamonds. I would come to I will just, in a couple of minutes, I will say why fluorescent nanodiamonds. For the moment, you just remember that these are fluorescent particles which can be used for live cell imaging. So we treated these neurons with fluorescent nanodiamonds, and within a couple of tens of minutes, we can see that these nanodiamonds are internalized into the neurons, and they are encapsulated into endolysosomal compartments, basically, because it's an endocytosis process that is going on with the nanoparticles. And then we tracked it for almost an hour using total internal reflection fluorescence microscopy, which gives this TERF microscopy is basically a wide field technique that would enable you to image at a better signal to noise ratio. So we image these neurons, and this is I'm going to play a movie. I hope you can all see the movie. Can somebody please confirm that? Do you see the?
Yes,
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 sir.
Nitin.
Thank you. So you can see these red vesicles moving, right? So these are nanodiamonds moving within the dendrites of the neuron. All what you see here are dendrites. I'm showing you a face image to show the neuronal structures because I wanted to highlight the nanodiamonds here. But we had confirmed that these structures are dendrites and not axons. So you can see that the nanodiamonds are moving there, right? And they move, if you closely observe, they move for some time and then they stop somewhere for some time and then again they move. So they have this peculiar feature of movements within the cargo trafficking is happening, right? Now, as I told you, we imaged this for like, we can image it for, even for an hour, but these images were taken for like 2 minutes or 3 minutes movies that we were doing. Now I will come to why we were using these nanodiamonds. Nanodiamonds are very bright. So what do you mean by brightness? So if you give, usually your dye molecules like DAPI or Alexa-Fluor dye molecules, the rhodamine and cysteine-based dyes, they bleach out very fast, right? If you shine them for, say, a few minutes itself, continuous irradiation for a few minutes, even with the minimum laser power, you will see them getting bleached off very fast. That is because there are several reactive oxygen species or other molecules that might bind to these dye molecules and change their orientation completely such that they will not be able to collect or they will not be able to absorb the light that you are giving. Unless they absorb the light, they cannot give you the fluorescence, right? Whereas nanodiamonds are crystals where defect centers are packed within the crystal structure. So no external ambient. So the crystal structure basically protects the fluorescent species very well, such that the external ambience like reactive oxygen species or any other molecules wouldn't, capture these fluorescence and push them to the dark state or let them bleach. So there is no blinking or bleaching behavior happening in nanodiamonds. That's one thing. The other thing about nanofluorescent nanodiamonds is that you can have almost, or the quantum efficiency of nanodiamonds is almost 1. That means you give a certain number of photons, you get almost the same number of photons as emission, fluorescence emission also. That's because the light is not lost in the form of heat or any other vibrational transition or energy loss. Most of the light you get back. So they have very high brightness and the brightness stays for longer time. So that enables you to do single molecule trafficking or single particle trafficking. Because you are trafficking, you are tracking these molecules at very fast rate. You also want to track them very fast and you want to track them for longer time. So these high efficient fluorescent particles such as nanodiamonds would enable you that. You may not be able to do this kind of experiments with this rate. We have imaged this at almost 50 millisecond per frame. That is all that's fast how we do this. We want to do it very fast at millisecond time scale resolution because these proteins, when they get trafficked, if you are taking one frame, the time interval between 2 frames is 1 second, for example, then you are losing every information that happened within that 1 second, right? Because these water proteins move at a rate of micrometer per second. That's the speed at which they move, micrometer per second. So these vesicles would definitely have a lot of details within 1 second. Within 1 second, they might have done a lot of things. They might have moved, they might have stopped for some time, they might have diffused for some time, they might be confined somewhere, and then again they went on for doing affected motion. So many biophysical features might have already happened within that 1 second time scale. So you always want to track them very fast at millisecond time scale, and also you want to track them for longer period. So that's where the fluorescent nanodiamonds come into the picture and enable you doing this. So here I've shown you how the brightness is basically going to help you, because this is the single molecule image. This probably is not an image from the nanodiamond that you're seeing here in this. It's any fluorescent molecule. And if you just draw a line across it, you will see the intensity curve, right? So you are seeing this mountain that you see in the next picture. So the photons are there on the y-axis and the position is there on the x and y. So each time in each frame, you're trying to locate the position of the molecule from the peak position that you get from the this mountain. So if your fluorescent molecule is very, very bright, that it has so are we going to take the questions at the end, or we take the questions in between? Because I saw a question just popped up.
We could take it at the end, Nitin, maybe.
Okay, all right, thank you. So if you see if the fluorescent molecule is not very bright, then the mountain, the peak of the mountain is going to be very flat. It is not going to be very sharp. So you will have, suppose you are climbing a mountain that is very hill, that is very flat, and you want to put a flag on the peak of the mountain, then you might have so many options to put the peak, put the flag, right, to find the peak. Whereas if your mountain is really, really, really sharp, and it has probably very few locations to put the flag and locate the peak of the mountain. So you want the mountain to be very sharp. That meaning that the signal to noise ratio should be very high. That means the number of photons that you collect compared to the background information or the background light should be very, very high. So nanodiamonds are very bright. So the error with which you are going to locate the position of the molecule is going to be minimal. At the same time, they are going to not be affected by the ambient conditions. So they will stay longer time, and so that you can track them for a longer period. So I hope I conveyed the message how different fluorophores can impact the error with which you are tracking your molecules in a live imaging session. All right. So very good. So you have used a fluorescent particle which is very bright to track for a longer period of time. You can get these kind of movies. So what, right? We can, just after taking the movies, we don't end the story there. We go into the analysis part of it to deduce the numbers out of it. So how do we deduce the numbers? That's how we do that's where we do the single particle trajectory analysis. So these, I'm showing a movie here again, where we have tracked the nanodiamonds in the dendrites for in a few minutes. You can see them going back and forth. These are different vesicles going back and forth, probably endosomes and lysosomes, multiple endosomes and lysosomes moving back and forth in a small region of a dendrite. And then what we do is, like, as I told you, at each and every frame, we are trying to record the position of the molecule at single molecule level or single particle level. So actually we try to, we can do some calibrations to see how many nanoparticles are within each endosome, and we calibrated it to be 1 or 2. So almost it is at a single particle level. So you get these positions of the track as you see here in this green curve. So I represented all the positions with so many dots. Now, the logic that we use behind the tracking is that we have to analyze these tracks to understand whether they are pausing there, or whether they are stopping there, or whether they are moving continuously without any problem. So we want to extract the features such as how much they move, how fast they move, did they pause somewhere, where did they pause, why did they pause, for how long they paused. All those biophysical features we want to extract. For that, we use this criteria called confinement ratio. So what does it mean? So imagine that the position, these are 4 different positions that I'm showing, 1, 2, and 3, and 4 in green dots, and the vectors in between, or the distance between 2 of them is denoted as D1, D2, and D3, and the displacement, that is from the first point to the last point, is given as capital D. Now if you take a ratio of displacement over the sum of the distances D1 plus D2 plus D3, and I mark it as R, if the particle was moving on a straight line, a perfect straight line, the ratio obviously is going to be 1, right? But at any instant, if the particle was not moving very fast in a straight line, rather it just got kind of a diffusive behavior, like it was moving like a zigzag and all those things. Then if you take the sum of d1 plus d2 plus d3, and if you take the ratio of the displacement from d1 to d3 over the sum of the distances, that is this ratio r, it is definitely it is going to be less than 1. Right, so this is the ratio that we call as confinement ratio, and we applied a condition that if the confinement ratio is above 0.8, we didn't want to be very strict about being 1, because no particle moves in a very straight line, because the microtubule can be a little bit twisted, or and then the motors can move around it or across it. So you will not be able to see a very straight motion, but you will see some directionality associated to it. That's why we put a ratio of 0.8, which is already a very strict ratio. So if the confinement ratio is above 0.8, the other condition that we applied was the angle criteria. So if something moves very straight, then the angle produced between them is almost 0 between the vectors. But as they start making these kind of angles between them, so if it is more than 90 between the angle between two vectors, then it's almost going to reverse its direction. So that is when we thought that, okay, using an angle criteria of more than 90 degrees would enable us to see if it is moving in the same direction, or whether they are reversing its direction, or whether it is going to diffuse out rather than continuing with its directed motion on the motors. And you might have already heard from Deepak about mean square displacement and how that can be used as a criteria for distinguishing between a diffusion versus a confined motion versus a directed motion mediated by motors, right? So from mean square displacement versus time curves, we can actually find out a diffusion coefficient. So if the diffusion coefficient is 1 or a little bit over 1, it is mostly diffusive in nature. The motion is diffusive in nature. If it is much larger than 1, for example, above 1.5, then you can conclude that the motion is no more diffusive. Maybe rather it is a motor-driven process. So that is where we put all these criteria like confinement ratio, angle condition, and diffusion coefficient above 1.45. So all these conditions were applied to each and every vector in this that contributed from these different points, and then we scanned through this trajectory with these conditions, and then we figured out that which of these points would be a confined one, and which of these more points would be diffusing, and which of these points would be actually contributing to a directed motion, right? So everything that you see here after the analysis, everything that you see here in green represent a directed motion because they followed this condition criterions, and whereas everything that you see in blue did not fall into this criteria, kind of telling you that maybe these regions in blue contributed to a pausing behavior. They just paused there for some time, or they discontinued their directed motion behavior or motor driven behavior, and the motor just stopped there for some reason. We don't know why. And then it may be diffusing there for some time, and then again it continues with its directed motion. That's what we deduce from this kind of an analysis. And then from these analysis, we can get these biophysical readouts, such as for how long the blue regions would give you information like how long they paused, how many times they paused, and from the run regions, that is the green regions, you might get what is their speed or the velocity with which these vesicles were moving, and what is the run length with which they are moving. By run length, biophysically, the run length means that the distance for which a motor is staying on the microtubule before it stalls off from the microtubule. But for our case, we have taken the run length to be the distance between two consecutive pausing behaviors. So for example, the distance from the first pause to the second pause, that is what we call as run length. And the time analog of the run length can be called as processivity of the motors. So these are the biophysical parameters that you can get out of the single particle trajectory analysis. Now we go and check for all those neuronal trafficking that we saw here, right? These are, as I told you, these are the movement of the vesicles within the dendrite. So we went and analyzed what is the passing time, what is the pause frequency or the velocity of all these vesicles that are moving in the neurons. And then we figured that we wanted to know how sensitive it is. So our objective was to understand whether this single particle trafficking is sensitive to the molecular changes happening, the protein aggregates or the molecular changes happening within the dendrites and the axons, right? So we looked into, we wanted to make sure that this is working. So there is a drug called nocodazole. Nocodazole depolymerizes the microtubule. So first we took the nocodazole and tried to take it at very small concentration, like 2 nanomolars and 5 nanomolars, 2 different concentrations, and try to treat it with the neurons. And then we saw here you can see the graph. This is for velocity, run length, processivity, and pausing time. The gray bars indicate the control situation. The green indicates the 2 nanomolar concentration, and the red indicates the 5 nanomolar concentration. So you could see that the velocity and the run length has decreased significantly with as you increase the nocodazole concentration, whereas the processivity also got decreased, whereas the pausing time, processivity, as I told you, is an analog of run length. So processivity, time analog of run length. So you will see that also getting decreased, whereas the pausing times have increased. So this already shows that, you know, even with such small change in the drug, you can already sense that with the high-end single particle tracking. Similarly, we wanted to relate it to the Alzheimer's protein, that these amyloid beta precursors, which eventually form oligomers. So we had these oligomers. We treated the cell with these oligomers, the neurons with the cell oligomers. The oligomers can form trimers and tetramers from the monomers. So if, depending on their concentration. So we tried to do 2 different concentrations. One is 0.5 micromolar, the other one was 1 micromolar. And you can see again the blue curve in the blue bars and the curves indicate the lower concentration. The magenta bars indicate the higher concentration of amyloid beta, and the control is in gray. You could see that even such small variabilities, the single particle tracking is very much sensitive to that, such that the velocity, run length, processivity, pausing time, all these biophysical readouts are going to give you differential values for the differential concentrations, right? So our experimental setup, our assay is working in relevance to Alzheimer's protein aggregates. That's what we figured. Then we went to look into another protein called MARC1. It's a kinase. What does this protein do? It phosphorylates the microtubule associated protein or MAP that binds to the microtubules on the dendrites. The same way tau binds to the microtubules in the axons, the MAP binds to the microtubules in the dendrites. So these experiments were done in the dendrites. So there were mouse models that were generated with overexpression levels of MARC protein. So you can see the differential level of the protein in overexpression of MARC in different mice models. So there was a line called line 38, which is the orange one is the wild type. The one of the lines, line 38, which is the heterozygote, had a very slight expression difference compared to the control level for the MARC1 protein, whereas line 8, which is another line of the mice that was generated, had a higher level of overexpression of MARC1 protein. So even with such small levels or slight variabilities of MARC1 protein, we could see that the velocity, run length, and the processivity had significant differences for the trafficking of nanodiamond containing vesicles. Why so? Because as I told you, the MARC1 protein phosphorylates MAP, and the phosphorylated MAP detaches from the microtubule. So when the motor arrives, if there are no MAPs decorating the microtubule, then the motor can pass without much hindrance, right? That's why you see with increasing level of MARC1 protein, you see the velocity and run length and the processivity going up. We couldn't see much of pausing behavior, unfortunately, from these results. That's why the pausing features are not included in here. So the same work has to be done for tau protein, which was not very convenient, not very successful in generating variabilities to such small levels of tau proteins in the mice like generated. So we couldn't do that either. But nonetheless, these experiments show that single particle tracking of endolisosomal, I missed the O here, endolisosomal compartment is sensitive to protein implicated in neurodegeneration. So we have shown that the take-home message is the trafficking readout reveals endophenotypes for neurodegenerative and psychiatric diseases. And the next question we want to ask is like, traffic readouts could be mapped to cytoskeletal organization. What will be the mechanism that enable these pausing behavior or these run behaviors that we saw? How can we correlate these biophysical parameters to the microtubule architecture itself? That's what we wanted to explore next, or the mechanism behind this. So coming back to the mechanism basically what I just mentioned was in the beginning of my talk, that this could be referred as molecular traffic rules, right? So the same way we have like stop, go, heavy vehicles move on certain roads and all that. Similarly, these molecular interactions with map, motors, and microtubules could also lead to different set of traffic rules. So what could those rules be? So in my imagination, it could be something like this. So if a lysosome the motility of the lysosome enabled by motors here, and it is moving on a microtubule decorated with the maps, right? But the maps, as we discussed, is in a dynamic relationship with the microtubules. So when a motor sees the map, maybe the map is giving a stop signal or a red signal to the motor. Or maybe it is asking the motor to go back, reverse its direction. Or maybe it is asking the motor to take a U-turn such that it can rotate over the microtubule and cross the barrier. And it's not only maps, there are other features on the microtubule, such as microtubule post-translational modifications. Microtubules are made from alpha and beta tubulin monomers. And these monomeric proteins, the amino acids of alpha and beta tubulin can be modified by different chemical groups such that post-translational modifications can happen to them. And these post-translational modifications can also contribute to all these traffic rules such that they can ask the motor to reverse, stop, rotate, or even switch tracks, take a bypass road or something like that. So now the question that we want to ask, like, do maps and microtubule post-translation modifications define or set specific molecular trafficking rules? So to understand that, first let me briefly tell you what are the major PTMs that we observe with tubulin. So here you have alpha tubulin and beta tubulin monomers. These alpha and beta tubulin monomers polymerize to form the microtubule. So the terminal tail of an alpha tubulin monomer has a tyrosine residue at the end. Okay, so this tyrosine residue can be cleaved off by the carboxypeptidase vasoheptanes. So that would make the alpha tubulin a D-tyrosinated tubulin, and the microtubules that contains the D-tyrosinated tubulin would eventually form the D-tyrosinated microtubules. So if there is tyrosine, then it is a tyrosinated microtubule. If the tyrosine is cleaved off, then it is a D-tyrosinated microtubule. Similarly, if the lysine residue at the 48th position is functionalized by an acetyl group, then you get an acetylated microtubule. If that functionalization is not there, then that chemical modification is not there, then you get a deacetylated microtubules. So these are the major post-translation modification that we observed in epithelial cells. But in neurons, you would also see polyglycylation, polyglutamylation, and all other modifications. These are like glycine residues being added to the terminal tail of alpha tubulin, or glutamate residues added to the terminal tail of beta tubulin. This is how the alpha and beta tubulin monomers can get modified, leading to different subsets of post-translationally modified microtubules. Now, in our epithelial cells, when we and antibodies are available against these different modifications. So when we try to stain for acetylated versus non-acetylated microtubule, this is what we saw. We see here, the acetylated tubulin in magenta, and in green you are seeing the deacetylated or the non-acetylated microtubules. Now you see clearly there are two tracks, right? Two different sets of microtubules. The question here is like, what kind of trafficking regulation, that traffic rule this might set, these different post-translational modifications? Would it be something similar to what we have in our daily life, right? If you see the image given at the right-hand corner, top corner, you see that there are tracks for pedestrians, there are tracks for cyclists, and there are tracks for heavy vehicles and light vehicles. So we have this kind of a scenario to avoid collisions, right, accidents, and efficient trafficking. So are these post-translation modifications in the microtubules offering different subsets of tracks, providing different routes for different tracks for different kind of motors? This is what we wanted to know. Basically, what we want to know, what we want to deduce is that when the vesicle traffics over an acetylated versus non-acetylated, or a detyrosinated versus a tyrosinated microtubule, are they going to be different? Whether they are going to post-mode, whether they're going to move faster, whether they're going to get reversed, whether they're going to get a U-turn, all those factors we wanted to know, and whether that has an implication and if this is not in order, then what is the implication in dysregulations that might lead to neurodegeneration? This is what we wanted to try. But before going into neurons, we wanted to check this mechanism in epithelial cells or in normal cell lines. And then once we have some conclusions, we would extend it to neurons. Otherwise, we will be sacrificing a lot of mice without solid purpose. So to understand the details, now to get to know how much of these different tracks are present and how they overlap with one another, all those details we want to understand from here, right? Before we go further, before we know how they track between two different differential trafficking of these vesicles in different microtubules, we want to get the details of this microtubule PTM architecture itself. For that, if you try to zoom in, if you digitally, one might think that, okay, I can just digitally zoom into this data and then figure out how they are organized, how they are entangled with one another. But as I zoom in, instead of getting more information, I'm losing the information, right? I'm just seeing a pixelated image here. This is because of the diffraction limit offered by the optics itself, the microscope itself. So there's a limit associated to your microscopy. So no matter how hard you try to zoom it digitally, you're still going to get a blurry pixelated image. And what is this diffraction limit basically mean? It means that if you are trying to image a fluorophore, you have tagged your protein of interest with a fluorophore. That fluorophore can be another protein like GFP or RFP, or a fluorescent dye molecule like Alexafluor or any other rhodamine-based dye molecule. These fluorophores basically come with a size of, say, few nanometers, 3 to 5 nanometers. But when you image them using a lens, their image would look as big as 200 to 300 nanometers. That's a basic problem. So you see, your molecule can be anywhere, your 3 nanometer fluorophore can be anywhere in this 300 nanometer space. So you're not able to get where it is actually located. The image looks as big as that. And how will it contribute to the diffraction limit, or how we are not able to resolve two molecules because of this? I'm coming to that. So focus on the left-hand side of the slide, what you're seeing. So here in red, I am showing a point source. The fluorophore is a point source which can radiate light in all directions. So this is your GFP or your dye molecule that you're trying to image. Here is a lens with a certain diameter D. And when this fluorophore light is collected by this lens, you will see diffraction patterns like this, concentric rings like this, right? And if you draw a line across this, you would see this intensity profile that is given above it, as we discussed for example. So the same image of this, and below that you are seeing the real image in a microscope that you would see for this fluorophore that you are trying to image. And you know that the central maxima of the diffraction pattern is represented as this mountain peak or the maximum intensity region, or in the real image it would look these orange pixels are the ones that is a central region of the image. So this central maxima has a definite size. It cannot be minimized beyond a level. That size is determined by an equation called lambda over 2 times the numerical aperture. So numerical aperture is the almost the light collection efficiency of the lens. If the numerical aperture is infinitely large, then you can reduce the size of this central maxima, or this intensity mountain will become very, very narrow. It will start becoming sharper and sharper and sharper and narrow. The width of this Gaussian is going to get very small. Also, if you increase the wavelength, right, then also you are going to get it narrow down, right? So either you have so if you normally in our optical system, the numerical aperture of most of the lenses are going to be of the order of 1.5. You cannot increase it more than that, at least for the moment. And since we are using fluorescence microscopy, it's a visible range. The wavelength is always in the visible range, right? So somewhere between 400 to 700 nanometers. So if you use a higher wavelength to get better resolution, then the best you can use is somewhere close to 600 to 700 nanometers, right? So imagine that you use 600 nanometer wavelength and the numerical aperture of 1.5, then the central maxima is going to be of the order of 300 nanometers, right? So the size of this central maxima, the size of the Gaussian width, full width at half maxima, is always going to be somewhere close to 200 to 300 nanometers. So if two fluorophores are sitting very close to each other, then you are within a range of 100 nanometers, then you are not going to resolve them as 2. Instead, you are only going to see them as 1. So this condition of meaning that there is a limit of your optics, that the way the light interacts with matter, that contributes to a physical limit of central maxima, size of the central maxima being lambda over 2Na, was formulated by Ernst Abbe. And once Abbe formulated this, Rayleigh developed a criteria such that if two such single fluorophores are sitting apart, much larger a distance, like 750 nanometer compared to their diffraction limits, imaging of the diffraction limited images, then you are probably going to resolve them. So here the Gaussian peaks for fluorophore number 1 is given in blue, and the Gaussian peak for the fluorophore number 2, the intensity profile, is given in red. So as they come closer, their intensity profiles are also going to come closer, right? The diffraction limited spots are also going to come closer. But till they are at this point, you can actually resolve them, and then they cross that, you wouldn't resolve. So what does this point mean? So this is the limit at which you can actually resolve something, and once they go beyond that, you will not be able to resolve. So Rayleigh said that if the central maxima, so this, if you look at this point, you see the blue curve has a central maxima, the central maxima of the blue curve and the first minima of the red curve actually overlaps. So the first minima of one fluorophore overlaps with the central maxima of the second fluorophore. That's when you stop resolving them. If they come beyond that, if they go beyond that, then you will not resolve them. But if they are above that, you probably would be able to resolve it. So as long as the central maxima of one fluorophore is away from the first minima of the second fluorophore, you will still be able to resolve them. But as they come beyond that point, you will not be able to resolve it. That is what Raleigh's criterion means. So with such a limitation, different optics, we have developed microscopes with different level of understanding this limit of diffraction and overcoming this, and to what extent we can use different optical system based on this, the resolution that it can offer you is summarized in this table. So if you are just looking into whole animal imaging or tissue imaging, you can always go for optical tomography or MRI, wide field depth microscopy and all that. If you are looking into single cells, bacteria, again confocal microscopy, structured illumination would help you. But here the question that we are trying to answer is at protein levels, right, which are of the order of 10 nanometers to few tens of nanometers, or vesicles or organelles which are of the order of few hundreds nanometers of the size. That's when super resolution microscopy came into the picture. So there are many methods by which you can break the diffraction limit and go beyond that to a few hundreds of nanometers, like stimulated emission depletion microscopy or single molecule localization microscopy. But I'm going to focus on the single molecule localization based microscopy. That's expertise that we have in our lab. So what is the concept of single molecule localization microscopy, and how are we going to overcome the diffraction limit with the help of single molecule localization microscopy? So you might all know that Nobel Prize in Chemistry was awarded in 2014 to Stephen L. Eric Betzig and William Moriner for different methods by which they enabled the breaking of diffraction limit. But we will stick to the concept of single molecule localization, which was formulated by Betzig. But this concept of single molecule localization was actually done by Paul Settig in with a technique called Fiona. And so here, as I discussed before, you have the image, you have the the intensity profile. If you are able to locate the peak of the mountain very accurately, then you are localizing where this molecule. So you consider this entire image to be a haystack, and you want to pinpoint the position of the molecule with the needle. So where you will put that needle is the question, right? The haystack is the entire 500 nanometer image that you get because of diffraction limit, and you're trying to see the intensity profile to say precisely what is the position of my molecule. And as I told you before, the error with which you are going to locate the position of the molecule depends on the signal to noise ratio. All of this error is given by sigma square xy, and all the factors like the photon count and other factors that contribute to the signal is in the denominator. All the factors that contribute to the background is in the numerator. So the more you reduce the background, the more you increase the signal, or in general, if you increase the signal to noise ratio, the sharper is the peak, and with less error, you detect the position of the molecule. That's the concept of single molecule localization. How can we use this in overcoming the diffraction limit? Now, I'm going to think that, okay, I have a lot of molecules overlapping with one another. Their central maximas are overlapping with one another. So that's why I'm not able to resolve. So if I go and locate their central maxima very precisely, then I should be able to resolve them. But as I look into this image, I can see that I'm not able to place the cross position, the central position, because molecules are all overlapping. So somehow I want to temporarily separate them. If I only had few of the molecules present here, which are non-normal, then I can probably locate their position very accurately. So the problem is that they are spatially overlapping, and the solution to this is you should temporarily separate them. If you did not understand that very clearly, we can demonstrate it right away. So I would request all of you to just unmute yourself. Did you all unmute yourself? Yeah. And now,
Yes.
Yeah. Now when I'm going to count 1, name. Okay. At the count of 3, all of you should together say your name. 1, 2, 3. 3.
Unless
Okay. But now you can all mute yourself. So you see, if all of us said our name at the same time, all your sound is going to overlap with one another. I will not be able to pick each name separately and locate your position, right? On the other hand, now you can mute yourself. Okay, at the count of 3, all of you should together say your name. Yeah, thank you. So, but if I ask only one of you or 2 of you to say your name one after the other, then I can always go and I will understand your name and I can also locate you, right? So that's the same thing. So I'm going to, there should be a way by which I should be able to temporarily separate these molecules. That's the key. So the idea behind STORM or palm imaging, which uses single molecule localization, goes like this. So here I'm showing any DNA or microtubule or anything that I want to image, and I have decorated them with this fluorophores. These yellow dots represent the fluorophores. For convenience, I have shown these fluorophores very sparse, but ideally when you try to do imaging, you would like to have the fluorophores decorating each and every point in your protein structure, right? So now if all these fluorophores are giving you 200 to 300 nanometer image, they are all going to overlap with one another. And that overlapping will give you a big blobby red image that you see here. So I know that the protein is there, but I'm not resolving its structure. Whereas if I temporarily separate them, meaning that I switch off all the fluorophores somehow, and I randomly switched on one of the fluorophores, then I can precisely locate its position and I mark its position. And then I switch it off, then I randomly switch on another fluorescent molecule. Again, I locate its position, right? So similarly, I can go ahead and do the localization for all the fluorophores one after the other and put together the localizations. Now, if I put together the localizations, I have reconstructed the image of my the structure that I wanted to image. And now the image is no more limited by diffraction. So this is the idea behind Storm and Pound. So this is how we do the experiment. So here,
Nitin, you have 10 more minutes to go.
okay.
Thanks.
So here you can see that the molecules are blinking. We have temporarily separated them, and then we are putting together localizations to reconstruct the image, right? So with this, how do we resolve it? So you can see the microtubules in a conventional imaging mode, like a normal TERF microscopy. And with the Storm, you can clearly resolve these structures much better. So you would appreciate it from the zoomed in images, like here the entangled microtubules were not able to be resolved in conventional imaging, whereas it could be clearly resolved in a super resolution Storm or single molecule Storm data. Now, moving on with this, we can you can also see that the previous problem that we had, we were not able to resolve these differential microtubules. Because of the post-translationally modified microtubules could be clearly resolved. Now one can see how they are entangled. So it's not just these pretty pictures, okay? We need to deduce quantitative information out of them. So you can binarize this with using images software. You can binarize this image, putting a threshold to their intensity, and then this is the binary image, and then you can calculate the area, that is how many pixels in this image is occupied by, and this how many pixels are occupied here with the binarized format, and then you can calculate the percentage of microtubule area in this image and this image, and then you basically understand what is the percentage of tyrosynated versus detyrosynated microtubule or acetylated versus non-acetylated microtubules. In that way, we have quantified the different distribution of different microtubule population in epithelial cells. We have calculated that tyrosynated microtubule contributes to about 65%, whereas detyrosynation is only 35%. Acetylation is about 70%, non-acetylation is about 30%, and acetylation and tyrosynation can coexist. I mean, detyrosynation and acetylation can coexist. So we figured that out of this 70% acetylated tubulin, about 55% is tyrosynated and 45% is detyrosynated. So putting all these values together, we deduced that in epithelial cells, the distribution of microtubule post-translation modification goes on like that. There is a 30% of acetylation in detyrosynated microtubules, 40% of acetylation and tyrosynated, and about 28% of non-acetylated and tyrosynated. Now the question is like how the endolysosomal vesicles are distributed in these different subset of microtubules, right? And how they lead to differential trafficking. For that, we did two-color Storm data. This image you are seeing is also a super resolution data. So here in green you have the lysosomes, and in magenta I am showing you the detyrosynated microtubules. So the arrows point towards the lysosomes that are associated with the detyrosynated microtubule, and the arrowheads represent the lysosomes that are away from the or not associated to the detyrosynated but associated to the tyrosynated microtubule. How do we estimate this? How do we quantify this? So in localization-based microscopy, we can actually find out the number of localizations from each of these points, okay, each of these images. So it's an analog to an intensity-based approach in conventional microscopy. Instead of intensity, we call it localizations. So you can put together the localization, the green localization and the magenta localizations. And if the localizations overlap with one another, then we think that when we estimate that this lysosome is associated to the microtubule. But if the localizations are separated by more than 50 nanometer, we put a condition of 50 nanometer because we from literature, we understand that the adapter proteins and the motor proteins that bind to the lysosome and then bind to the microtubule would contribute to almost 50 nanometer. So that's why we put a condition of 50 nanometer between the localizations. And if it is beyond that 50 nanometer, then we probably do not associate this lysosome to be walking on this detyrosinated microtubule. So with such a quantitative approach, we figured that there is an enrichment of lysosomes on detyrosinated microtubule because we are trying to see the percentage of organelles on percentage of microtubules. Since detyrosinated microtubule is only 35%, if about 70% of the lysosome associated to 35% of the detyrosinated microtubule, that shows there's a twofold enrichment. So if this enrichment ratio is 1, that means the distribution is even. The enrichment is over 1, then the distribution, you estimate that there is an enrichment, it's not even distribution. So for lysosome, we see that the enrichment is on detyrosinated microtubule. On an acetylated is evenly distributed. We see the same enrichment for autophagosome and autolysosomes. And mitochondria, we didn't see any such behavior. Now, coming to the, so now we see that there is these images are okay, but we want to do live imaging, right? But STORM is a fixed cell technique. You have to immunostain to get these super resolved images. So you do not get any dynamic information. So I combined what I did in my first part of the talk where we do single particle tracking. So we first do the single particle tracking where you get almost 50 millisecond time scale resolution. We can track these lysosomes on these cells. Then we immediately fix the cell and then do the super resolution imaging to get the highly spatially resolved microtubule structure. So from single particle, you have temporal resolution of 100 milliseconds. And from STORM image, you have the 20 nanometer spatial resolution. Then you put together, then you clearly know on which microtubule the lysosome is moving and how they are taking turns around and whether they are pausing and all that. So we basically have high spatial and temporal resolution. So like that, we track the lysosomes first, then we figured the STORM, the microtubule tyrosination, detyrosination from the STORM imaging. We put together the tracks on these different microtubules. Now we clearly know which lysosome move on tyrosinated and which moves on detyrosinated. Then we do again the particle trajectory analysis to understand the run and pause behavior. We will calculate the motility parameters like speed, run length. We figured that we quantified that the speed and the run length reduces as it moves on detyrosinated microtubule, while the pausing behavior such as number of pauses, pausing frequency, and pause time increases as it moves on a detyrosinated microtubule. Along with this data, we also saw that out of all the fraction of lysosome that is enriched on a detyrosinated microtubule, most of them are immobile. So these two data, combining these data, it suggests that lysosomes are most paused, or their interaction or their motility is hindered on the detyrosinated microtubule. So we thought that maybe they are interacting with their autophagic counterpart, the autophagosomes. Exactly, that's what we found. Here you see again correlative microscopy. In white you are seeing the detyrosinated microtubule. In magenta you have lysosomes, and in green you have autophagosomes. You should know that I have only labeled for the detyrosinated microtubule here. All these empty spaces that you see should be decorated by the tyrosinated microtubule. But if you see the lyso-autophagosomal fusion majorly is happening on the detyrosinated microtubule. So you can see that clearly from this movie where the autophagosome is moving on a detyrosinated microtubule and merging with the lysosome that is already present on the detyrosinated microtubule. I play that one more time. The green autophagosome moves on the white detyrosinated microtubule to merge with the magenta lysosomes. Now we try to eliminate these detyrosinated microtubules by knockout treatment, and then all the microtubules will be eliminated by knockout treatment. You recover them with the parthenolide is a drug that inhibits the vasohilin enzymes that promote detyrosation. So in this recovery process, acetylated microtubules are recovered, but detyrosinated microtubules are still eliminated. So we try to look into the autophagosome population versus the fused autolysosomal population using this tandem sensor of LC3B GFP RFP. So you see that the autolysosomal population, which is a fused population of autophagosome and lysosome, is significantly reduced if you take away the D-tyrosine microtubule. These experiments were complemented by protein overexpression experiments, and with tubulin tyrosine ligase overexpression increases the level of tyrosine and decreases the level of D-tyrosine. Similarly, inhibiting vasogibin expression by shRNAs, we also decrease the level of D-tyrosine microtubule. All these three experiments together told us that the population of autolysosomal, that means the fusion of autophagosome and lysosome, is compromised when you eliminate D-tyrosine microtubules. So that meaning that D-tyrosine microtubule is important. Then we looked into the motors that are responsible for these processes. Like we knock down the level of these motor proteins, kinesin 1, 2, and 3, one after the other. So we figured that the enrichment of the lysosome on the D-tyrosine microtubule was unaffected when you take away KIF3A and KIF1B. But when you knock down the levels of KIF5B, you see that the enrichment of lysosome on the D-tyrosine microtubule goes down. Similarly, the autophagosome and lysosome fusion contributing to autolysosomal population also reduced if you inhibit kinesin 1, that is KIF5B. And overexpressing the KIF5B knockdown rescued that phenotype, or we could see the autolysosomal population re-established in that phenotype. So together concluding that KIF5B somehow contributed to the localization of these lysosomes on the D-tyrosine microtubule. So our proposed model is that lysosomes are enriched on D-tyrosine microtubule, KIF5B localizes the lysosomes, and lysosomal motion is hindered, and that enables the lysosomal autophagosomal interactions. And D-tyrosine microtubules contribute significantly to the autophagy in general. Now, as a future perspective, we deduced some understanding about the microtubule PTMs in how they regulate the molecular trafficking, right? We also want to look, our future perspectives include how MAPs contribute to this kind of molecular trafficking. And we also know that autophagy is one way to eliminate these microtubule associated protein aggregation leading to neurodegeneration. It is being proposed that enhancing autophagy can actually lead to clearance of these aggregates. So knowing more from these basic traffic signaling, we can enhance, because autophagy is also regulated by microtubules. So we can enhance these clearance of aggregation and thereby rescue neurodegenerative phenotypes. And we can also identify the neurodegenerative phenotypes much earlier than they form plaques and tangles. With that, I conclude my talk, and I would like to thank Melike Lakadamayari and her group at Peregrine School of Medicine, where I did my postdoc. So all the work that I presented today are from my postdoctoral work I did with Melike at UPenn, as well as my postdoctoral work from at JNCASR, France. So our lab is still trying to stand on its feet. We have just started our experiments very little. And on the right-hand side, you see our lab at IIT Kanpur and its members. And stay tuned to have the updates on what we do in terms of autophagy as an approach for clearance of aggregates in neurodegenerative disorders. With that, I will be happy to take more questions.
Thanks, Nitin, for the wonderful talk. I think we'll take some questions, guys.
Yeah, sure.
Yeah, so the first question is the fluorescent nanodiamond tagged to a protein in the dendrites?
No, we just, we incubated these nanodiamonds with the neurons so that they go into the endosomal compartments. We figured that they are undergoing endocytosis by marking it with endosomal proteins like RAB5 and RAB-associated proteins and clathrin-mediated proteins and all those things. So we figured that it's an endocytic pathway, and the nanodiamond is encapsulated in endolysosomal compartment. So everything that you see is either endosome or lysosome moving in those neurons. But actually, you can tag proteins with nanodiamonds, but you have to choose very small nanodiamonds like 10 nanometer diamonds. But unless your protein is bigger than that, it will be difficult because these crystals can actually change the orientation of your protein itself. So it's a tricky business there.
Thank you, sir. The next question is, what is the molecular nature of the nanodiamond?
It's a crystal. It's a tetrahedral structure of carbon allotrope. Carbon has two allotropes. One is graphene and the other one is diamond. So it's a crystal lattice. It's a tetrahedral crystal lattice, and it's ideally diamond crystal should be all carbon, like the tetrahedral arrangement. All the positions should be carbon, but nature is abundant with nitrogen. So some of the carbon will be replaced by nitrogen. And this nitrogen and other defects within the crystal can come together to form nitrogen defect centers or nitrogen vacancy defect centers. And that contributes to the fluorescent properties. So it doesn't have any other biomolecular features. But you can definitely modify the surface of these nanodiamonds with functional groups like DSA or dextran or any other linkers that you want to put in so that you can attach other proteins and biomolecules of your interest onto these nanodiamonds.
Thank you, sir. Another question is, can we use nanodiamonds to track any other vesicles or characterize amount of protein?
You can. I think nanodiamonds are very good, they have very good sensitivity. The surface features can be modified such that it can be used to capture the proteins. So if you are using this in vitro assays, there are many studies. If you are interested, you can look into those papers. I have not done that, but there are people who do that. I know people who do that. So there are many studies. They can be used for capturing proteins and quantifying them in vitro. In vivo, using nanodiamonds for any vesicles, you can have localization signals on top of decorating the nanodiamond surface with specific localization moieties, such that it will specifically get targeted to, say, mitochondria or Golgi vesicles or things like that. That could be also done. Any other questions?
Hello, so the next question is, how are these parameters computed using software? I think Snigdha is trying to ask about the parameters of motor protein movement like run length and speed, etc.
So as we discussed, these are the different conditions that we applied, and once you get the position of these molecules, right? Position of this, once you have trajectories like this, these are like X, Y coordinates against time frames. So you can write so this, whatever I showed you here is basically a custom generated, custom made software that we apply all these conditions and then try to, if you have position coordinates and if you have time, then you can actually calculate all these parameters like speed, run length, velocity. Speed is nothing but distance over time, right? And you can calculate all those things. But even if you don't, if you're interested, you can always contact my lab and lab members. We will be happy to share with you these algorithms to analyze your data. But you can also use online already available plugins in Image2Numbers to calculate these parameters. So in the afternoon, our lab members Sanket and Jagan will guide you through how to use already available plugins to deduce these same information. But there will be limitations to those techniques. Like some of the precise informations may not be able to be deduced. So here we apply a lot of conditions to get very precise information with single particle tracking. So if you're not satisfied with chymographs or TrackMate that Sanket and Jagan will explain to you, you can always contact us and we will help you analyze your data with this algorithm.
Thank you, sir. The next question is, does single molecule tracking and further analysis have any advantage over chymograph generation and further analysis?
Yes. So my previous answer to my previous question actually answers this too, because chymograph and TrackMate, when Sanket and Jagan will tell you their advantages and disadvantages. So, you know, single particle trajectory basic with high efficiency will help you understand point by point trafficking. This kind of an algorithm that I'm showing you here will enable you to understand what is the feature point by point, but chymograph and TrackMate probably will not give you that kind of a precision data. It might take the entire track as such, and then it will tell you whether it is a confined motion, whether it is an anterograde motion, whether it is a retrograde motion, all those things. To deduce features within a track, you need to use this kind of analysis method.
Thank you, sir. Next question is, what is the size of the nanodiamond?
The nanodiamond size usually ranges from 5 to 10 nanometers to 100 to 200 nanometers. So you can choose based on your application. So if you are doing protein tagging and stuff, you want it to be as small as say 5 to 10 nanometers, but then their brightness will go down a little. If you're doing vesicle trafficking like this, you can use 50 to 100 nanometer because your organelle itself is going to be few hundred nanometers. So you can use 50 nanometer size. So the one I'm using is 50 nanometer. For the data that I showed you for neuronal trafficking, those were 50 nanometer average size.
Thank you, sir. The next question is, are there distinct populations of tyrosinated or detyrosinated microtubules? Or do you think these PTMs can alter reverse temporally?
Yeah, so as we told that there are distinct, these populations of tyrosination and detyrosination are very distinct from one another, right? You can see the magenta and the green never overlap with each other. They are very distinct. Whereas when you project it with total tubulin, they might overlap. As you see here, alpha tubulin is here, and projecting alpha tubulin along with acetylated, you see the color is white because they are overlapping. Whereas tyrosination and detyrosination, the microtubule can be either tyrosinated or detyrosinated. It cannot be both. Either the tyrosine residue is there or it is not there. So they are very distinct population, and we clearly quantified the percentage in epithelial cells. We are yet to do this in neurons. It's a bit more challenging over there because neurons, microtubules are much more packed than as radial as you see in epithelial cells. So the second question was what? Whether they temporarily, whether there is a dynamic nature associated to it, whether they, yes, because this process is reversible. The tyrosination, the tubulin tyrosine ligase will always put back the tyrosine at the terminal tail, and the carboxypeptidase vasohibin will cleave off the tyrosine from the terminal tail. Right. So this is a reversible process. So depending on how the enzyme acts, which enzyme is more, at certain different stages of the cell, probably different enzymes are upregulated and downregulated, modulated, such that the level of tyrosination, detyrosination can vary.
Alice is asking, what concentration of these nanodiamonds was used for neurons?
I think it was like less than few micrograms per ml. Like I think we use 0.05 microgram per ml in a well-confluent primary cell culture. Yeah, because we wanted it to be at single particle level, so we use very, very low concentration, microgram per ml.
Thank you, sir. Anubhav asks, how can a nanodiamond be cross-linked to a protein of interest in vivo?
Yeah, as I told you, like you can either use this you know, the, the surface of the nanodiamonds can be functionalized with any moieties that would link to proteins. So if you want to just bulk, you want to accommodate a lot of proteins and just do a bulk association of proteins onto the nanodiamonds, you can use BSA as one method. The other one is this biotin streptavidin conjugations that you do to just specifically conjugate molecules. That can be also done with the surface functionalization of the nanodiamonds.
Thank you, sir. The next question is, in cells that lack these post-translational modification, for example, D-tyrosination signatures on microtubules of HeLa cells, do you think the other PTMs would come into picture, or it is the level of the same modification which plays a role in trafficking?
Yeah, that's a very interesting question. So not all cells have the same level of different post-translational modification of microtubules, right? Different tissues, different cells will have different level of PTMs. Interestingly, we have seen that hepatocytes, right? HUH7 cells do not have acetylated microtubules, whereas they have considerable level of D-tyrosynated microtubules. So we are yet to explore whether the role of acetylation would be taken over by a D-tyrosynation there. We are yet to see that. Also, in addition to that, HeLa cells do not have D-tyrosynated microtubules. They have plenty of acetylated microtubules. And studies from Juan Bonifacino's lab have shown that same kinesin 1 motor, that is KIF5B motor, is hindered on acetylated microtubules. So probably in different cells, depending on the function, you know, what is the purpose there at that point? Everything is just coordinated spatially and temporally such that their function is performed. So these are very dynamic things, and it will have differential effect on different tissues for sure. And their function can be modulated based on where they are present and at what time they are present.
Thank you, sir. The next question is, in non-disease condition, the phosphorylation of tau may increase run length and reduce pause time. And second question is, do you think PTM alters in neurons of patients more specific do have ratio data between tyrosine to D-tyrosine and same for aceto?
I think the data in neurons are still emerging. We don't have a clear picture yet, but there are plenty of very recent studies have shown that there is a differential level of acetylation and D-tyrosination in dendrites versus axons, and that contributes to the sorting, how the vesicles get sorted into dendrites and axons. These differential population of acetylation and D-tyrosination in dendrites and axons can have an impact there. But the real implications in diseases and real functional relevances, those studies are yet to come. And I forgot the first question. Can you repeat the first part of the question?
Yes, sir. It's saying in non-disease condition, the phosphorylation of tau may increase run length and reduce pause time. Is it so?
Phosphorylation of tau may increase run length because in a healthy condition, non-disease, healthy condition, you know, there could be a balance at which the phosphorylation of tau is happening, right? So you may not, so you can only compare what is happening at a healthy, a disease condition with a healthy condition. So if you see that, you know, there is a nominal level of phosphorylation of tau, and that would lead to a specific set of run length, then that would be altered with a hyperphosphorylated tau, such that hyperphosphorylation of tau would lead to larger run length. That's what we would anticipate. But who knows? We might see something different. And maybe that's because some other protein came into the picture that would stop the motor from running, even though the tau was gone. But we have to do the experiments to understand that. But that would be a possibility. What the person suggested could be a possibility.
Thank you, sir. We will take one last question, which is on YouTube Live. I think it says that as we activate one particle at a time in STORM, does the time difference between two activations cause error, especially if the structures in the sample are moving very fast?
Very good question. So you don't want the molecule to stay on for a long time, right? Even if you are making the molecules blink, if one molecule showed up, if I asked you to tell your names at the same time, and then I told that that's a problem, and then I asked you to say one after the other. But if the second person says his name before the first person has finished saying the name, then again there is an overlap. So similarly, if a fluorophore blinks, it shows up and it stays on for a longer time, and within that time another molecule just showed up, then you are again not be able to resolve them very properly. So we need the molecules to blink as fast as possible, so almost at the level of milliseconds, so that we can get very sparse localizations, separated localizations, and if not, again the resolution will be affected. We will not get very good resolution if the molecules do not blink very fast. So the time factor involved in blinking of molecules is important. It should be very fast.
Thank you very much, sir. I think we have covered all the questions from participants.
Great. Thank you. So I can stop sharing, right?
Thank you very much, Nitin, for a wonderful session.
Thank you very much. I think that here the power is gone. I think I might be disconnected anytime.
Okay. So over to you, Akshaya, if you have any instructions for the afternoon session. Thanks everybody for the good questions and
For any further questions, if you have for all the participants, you can directly write it to Dr. Nitin, and he will be happy to take it. And for the afternoon sessions, the mail which we have sent yesterday, which contains all the instructions related to the plugin and the data set, kindly download that before the afternoon session. And we will be starting the afternoon session at 2, so we can break for lunch now. Thank you so much, Dr. Nitin. It was a pleasure having you on board, and thank you for putting it together for us. Thank you.
Thank you very much for having me, and good luck to Sanket and Jagan for the afternoon session, and all the participants. I hope you will enjoy that session too and get more clarity on single particle tracking with commercially available plugins. So thank you, Ravi and the team once again. Thank you all. Bye.
thanks.
How to log off?